All right, BIMM 100 students, the final is right around the corner. So in this session, we are going to do a super high-yield, focused review of Lecture 10. We're talking everything you need to know about pre-mRNA processing to just completely nail those exam questions. So here's our game plan. We're going to tackle the three huge processing events: capping, polyadenylation, and splicing. Then we'll zoom out and see how they all connect, and we'll wrap up with some pro tips that are specifically designed for Professor Pasquinelli's style of exam. Okay, let's dive right in. Lecture 10 is really all about taking that raw, brand new pre-mRNA transcript and turning it into a mature, stable messenger RNA that's actually ready for translation. You can kind of think of it like raw footage from a movie. It needs to be edited, stabilized, maybe given a title sequence before you can show it to anyone. Well, that's exactly what these three modifications do for our mRNA. We're going to start at the very, very beginning. Literally, at the 5' end of the transcript, and we're talking about the 5' cap. This is the first thing that happens, and believe me, it is a huge deal. So what even is it? Well, it's this special guanine nucleotide. It gets a little methyl group stuck on it, and then it's added to the very front of the pre-mRNA. Now, here's the key detail, and this is a total classic exam kind of detail. It's added in this weird backwards 5' to 5' linkage. This is completely different from all the other connections in an RNA chain, and that's what turns the cap into this special protective helmet, you know? It shields the RNA from enzymes that would just love to chew it up from that 5' end. The 5' cap has three absolutely essential jobs, and a great way to remember them is with 3 Ps: protection, passport, and promotion. So it's a helmet that provides protection from getting destroyed, it's a passport that gets it stamped for approval to exit the nucleus, and it's a promotion handshake that the ribosome recognizes to kick off translation. I would bet money that at least one question on your final is going to test you on these functions. So that brings us to the big question. How are you going to get tested on this? Well, Professor Pasquinelli loves these what-if scenarios. You're going to get some kind of mutation, some loss of function in a piece of the machinery, and you'll have to predict what happens next. So let's take a look at this question, pulled straight from your homework. It's asking, okay, what happens if the enzyme that slaps that critical methyl group onto the cap is just gone? What's the downstream effect? This is not just some random hypothetical. This is exactly the kind of critical thinking you'll need to do on the final. And this slide right here lays out the exact thought process you knew to follow. Step 1, identify the core problem. The 5' cap is incomplete. Step 2, remember its jobs. Okay, so protection, export, and translation are all going to be messed up. The final conclusion, you're going to see a massive drop in gene expression for pretty much all protein-coding genes. Okay, let's head over to the other end of the mRNA and check out the 3' poly-A tail. This modification is all about giving the mRNA a longer shelf life in signaling that transcription is done. Polyadenylation is, well, it's exactly what it sounds like, adding a long tail of adenine bases. But here's a super important detail for the exam. This tail is not coded for in the DNA. It's not part of the original blueprint. It's more like adding a long stabilizing streamer to a kite after you've already built it. There's a special enzyme, poly-A polymerase, that just adds it on after the fact. And this whole process is this precise two-step operation. First, there's a signal sequence in the RNA itself, that classic AAUUA, and it basically acts like a landing strip for the processing machinery. That machinery lands, cuts the transcript, and then poly-A polymerase comes in and just starts adding that long tail, 1A at a time. And that tail is absolutely vital for both stability and for getting efficient translation later on. And here we go again, another classic exam-style question. What happens if you mutate one of those other signals, that GU-rich element that helps the cleavage machinery find its spot? Well, the consequence is pretty straightforward. If you break that signal, the machinery never gets recruited. It's like a train just blowing right past its stop because the signal on the track is broken. Transcription just keeps chugging along, and you end up with an abnormally long 3' UTR that really messes with the mRNA's stability and translation. And the end result is you get a lot less functional protein made from that gene. All right, now for the process that really truly sculpts the final message: splicing. This is where the cell does its major editing job, cutting out all the non-coding junk and stitching the important parts back together. This is a core concept you have got to nail down. Introns are the intervening sequences. They get cut out. Exons are the expressed sequences. They get joined together to form the mature mRNA that actually codes for the protein. So, Introns in, exons out. Wait, no, scratch that. It's the other way around. Introns are out, exons stay in. Got it? Now, the exam is not just going to ask you to define an intron. That's too easy. Take a look at this question from a past exam. It's about a single nucleotide change that messes up something called an exon splice enhancer, or ESE. This is a higher level concept, and it focuses on the regulation of splicing, which is a favorite topic. So what on earth is an ESE? Well, think of it as a short sequence of DNA that's located inside an exon, and it acts like a bright flashing neon sign. It attracts these special proteins called SR proteins, and they act like landing lights, guiding the main splicing machine, the spliceosome, and telling it, hey, over here, this part is important. Make sure you include this in the final mRNA. So what happens if that neon sign breaks? Well, if you see an exam question about an ESC mutation, your brain should immediately connect the dots. The ESC is broken, so the SR proteins can't bind. The spliceosome doesn't recognize the exon, and poof, it gets skipped. This creates a shorter mRNA, which is going to produce a messed up, non-functional protein, just like in that RAS gene example from the old exam. Okay, so we've talked about capping, polyadenylation, and splicing like they're all separate events. But the real magic, the really cool part, is that they don't happen one after the other. It's time to tie it all together. And this brings us to this big idea called co-transcriptional processing. Basically, the cell is the ultimate multitasker. It doesn't wait for the entire RNA transcript to be printed out before it starts editing. No way. It's much more like an assembly line, where the capping, the splicing, and the tailing are all happening in real time as the RNA molecule is literally still emerging from the RNA polymerase. So who's the master coordinator of this whole assembly line? It's the C-terminal domain, or CTD, of RNA polymerase II. You should think of this long, flexible tail as a kind of programmable tool belt. As transcription gets going, these chemical tags, phosphates, get added to the CTD. And this changing pattern of tags acts like a signal, telling the tool belt which tool to grab next. First, it recruits the capping enzymes. Then it grabs the splicing machinery. And finally, it brings in the polyadenylation factors, making sure everything happens in the perfect order at the perfect time. All right, let's wrap this thing up with a rapid-fire review of the key strategies and the absolute must-know takeaways for the Lecture 10 material on your final exam. This right here, this is your ultimate checklist. You need to know the jobs of the cap, the tail, and the spliceosome. You have to be ready for those what if this gets mutated questions. And always, always trace the problem from the tiny molecular defect all the way to the final impact on gene expression. And don't forget, the CTD is the master coordinator. If you can confidently explain every one of these points, you are in fantastic shape. And I'm going to leave you with one final thought to chew on for the exam. Look, capping and adding a poly A tail are pretty much mandatory for all mRNAs. But which of these three processes gives the cell the most flexibility to mix and match pieces to create a huge diversity of different proteins all from a single gene? That's where the real regulatory power is. Think about that one. Good luck studying. You've got this.